Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 6.4, translation. In 6.3, we discussed transcription, which is creating an mRNA molecule out of DNA. And today we'll be discussing translation, which is when that same mRNA molecule can code for a protein. And we'll be talking about the different steps of translation and retroviruses. And so our suggested scale of visual representations 2D represent relationships within biological models, including mathematical models, diagrams, flowcharts, and systems, and argumentation 6E predict the causes or effects of a change in or disruption to one or more components in a biological system. And our biological system here is going to be the central dogma, which takes us from DNA to transcription to translation. Once again, gene expression is the process by which DNA directs the synthesis of proteins. So how can we code how can DNA code for your outward appearance and the functions of your body? By synthesizing the necessary proteins. And so we discussed in 6.3 transcription where mRNA is produced that carries the genetic message from the DNA, from the nucleus to ribosomes, which can then produce proteins which are either free in the cytoplasm or located on the rough endoplasmic reticulum. And so translation is going to occur now. This is the production of the polypeptide chain, a protein, using the mRNA transcript, and that of course occurs at ribosomes. And transcription and translation occur at the same time in prokaryotes. There is no RNA processing in prokaryotes like we talked about in 6.3. In order to cover translation, we need to discuss two other types of RNA. We discussed mRNA, which is messenger RNA, that carries the genetic message from the nucleus to ribosomes. But we also need to know tRNA, transfer RNA, which transfers amino acids from the cytoplasm to the ribosome to assemble a protein. So essentially how tRNA is going to work is it's going to bring a certain protein based on the base pair, and that's going to then contribute to our polypeptide chain, to our protein chain. And it has an anticodon that allows it to base pair to the codon on the mRNA. So a codon is a group of 3 nucleotides, and when the mRNA is exported, it's going to be read by the ribosome in 3 nucleotides. And based on the groups of 3 nucleotides, certain tRNA molecules are going to bring different amino acids that are going to then contribute to a different overall protein that's been coded for. So a group of 3 nucleotides codes for 1 amino acid. And ribosomes are made up of rRNA and proteins, a ribosomal RNA, and they have 3 binding sites for tRNA molecules. The first site, the P site, is going to hold the tRNA that carries the growing peptide chain. The A site holds the tRNA with amino acid that will be added to the chain next, and the E is the exit site for tRNA that have already dropped off their amino acid. So over here you can see our A site. That's where our tRNA is going to bind to the codon with the anticodon. It's got the opposite base pairs of whatever's seen on the mRNA, and it's carrying the amino acid. Then in the P site, we're going to be holding the growing amino acid protein chain, and the exit site is where tRNA can leave the ribosome in order to then repeat the process and bring another amino acid in. And we'll look at all of these steps in more detail. First up is initiation. A small ribosomal subunit is going to bind to the mRNA so that the start codon, AUG, is placed in the right position. All transcription and translation sequences are going to start with the sequence of AUG. That's going to begin protein production. And so the tRNA with UAC, methionine, is going to bond to the first codon. And UAC, those are going to be the base pairs to AUG. Adenine to uracil, uracil to adenine, and guanine to cytosine. And so it's going to bind the first amino acid, methionine, to the first codon. And then the large ribosomal subunit attaches, which is then going to allow the EP and A sites to exist. So we start with just the small subunit binding our methionine and our very first start codon, and then we have our large subunit that's going to allow for the growth of this polypeptide chain. So then the amino acid chain is going to elongate. tRNA with the correct anticodon is going to bind to the A site. So the tRNA that has the right 3 nucleotides that are base pairs with the mRNA. And so the peptide bond will become formed. It's going to move over to the tRNA in the A site, and then they all shift right. So what's going to happen is, you see, we start with our methionine, then the next amino acid is going to arrive by the A site. The original amino acid is going to bind to so methionine is going to bind to the amino acid on the A site, and then they're all going to shift right. So now the second tRNA molecule is in the P site, holding the growing polypeptide chain. The first one that carried the methionine can exit the ribosome, and a third tRNA molecule holding another amino acid can now enter the cell through the A site. And this process will continue until there's going to be the ending of this amino acid chain. So when a stop codon in mRNA is reached, and there are three different options for stop codons, a release factor protein is going to bind to the stop codon and the polypeptide is freed from the ribosome. And it's going to undergo some protein folding, like we've discussed when we discuss proteins and enzymes. There's going to be a signal peptide, which is a sequence of 20 amino acids that determines what the destination of the protein is. So what is its function, where is it going to go? And then the protein will finally be complete through the process of translation. And so what often is used to determine what kind of protein is going to be created is a codon chart, as you can see down here. So the question is, what amino acid will a DNA sequence of GTA code for? So when we begin with DNA sequence, we have to follow the central dogma. First, we have to transcribe the DNA so it can become RNA. And GTA, when we look at our base pairs, is guanine to cytosine, thymine to adenine, and adenine to uracil, because we're now looking at RNA, so we cannot use thymine as our base pair. So here's our RNA, but we don't need RNA. Now we have to go one further to find the anticodon on the tRNA molecule. So once again, do some more base pairs. Cytosine goes to guanine, adenine goes to uracil, and uracil goes to adenine. So our tRNA codon is going to be GUA. And now we can determine what amino acid we're looking at by using our codon chart. First letter is going to be G, so we go to our section here in the chart that has the G's. That's this row right here. Second letter is U, so we're looking at the first column right here. So now we're down in this box. And lastly, we have our third letter, which is A, adenine, and that's this third row. GUA codes for the amino acid valine. So it's key to remember that there are multiple stages of base pairing here to find the amino acid that you want. It is not just going from DNA to RNA. You have to go from DNA to the mRNA that through transcription, and then to tRNA through translation, meaning there are multiple rounds of base pairing. And anytime we're working with RNA, then we don't have a thymine base pair, we have a uracil base pair. But there is also an alternate flow of genetic information when we look at retroviruses, which have genetic information stored not as DNA, but as RNA. And so when the virus infects the host cell, as we can see right over here, this viral RNA undergoes a change through reverse transcriptase. It's going to become DNA, which can then integrate itself into the host cell genome. And once it's in the host cell genome, it'll be transcribed and translated as following the central dogma. So it's going to become mRNA, and then eventually this will result in the creation of more viruses. And that is how these viruses, quote unquote, reproduce. By infecting the host and then undergoing this reverse transcriptase to make their genome compatible so that they can then be transcribed and translated. So you need to know the steps of translation and the roles of rRNA and tRNA in translation, and also the differences in gene expression in prokaryotes where there is no mRNA processing and this happens almost simultaneously, eukaryotes where we have these laid out steps, and viruses where genetic information starts as RNA and undergoes a change to become DNA.